My name is Daniel Maitethia Memel, and my PhD project was titled Development of Photothermal-Induced Optical Scattering Modulation Technique for Malaria Diagnosis. My supervisors are Abdara Merenga from the Department of Physics in Kenyatta University and Professor Shira Wamaina from the Department of Electrical and Electronics Engineering in Dedan Kimathi University of Technology. So my presentation outline is as shown. So a molecule can interact with the light in a number of processes, including scattering, absorption, and polarization. When absorption takes place, a molecule is excited and goes to excited state, and after some time it relaxes and goes to ground state. And there are two main mechanisms that can happen, either when the molecule absorbs a secondary photon through a process we call radiative relaxation, or when the molecule emits it through a process we call non-radiative relaxation. Non-radiative relaxation gives forth to photothermal processes, which could include photoacoustic effect or thermal lens effect. So malaria is caused by Plasmodium parasites, which are transmitted by mosquito. So when a malaria carrying mosquito bites a human host, it launches the parasites in form of sporozoites into the bloodstream, and the sporozoites are transported to the river where they incubated, and after a while they are released again into the bloodstream where they attack red blood cells. And while in the red blood cell, they ingest hemoglobin and multiply, and at some point when they multiply within the infected red blood cell, the blood cell would rupture and the parasites would be released again into the bloodstream where they attack more red blood cells. And when a mosquito comes now for a blood meal again, it can carry a number of these parasites in form of merozoites and repeat the transmission cycle. So when the parasite is degrading hemoglobin, it breaks the molecule into two parts. The globin part, which they use for their nutrition need, and the heme part, which happens to be toxic to them. And so they have an innate mechanism of converting heme to an inert molecule called hemozoin, which is deposited in their food vacuum. And hemozoin absorbs optical radiation, and it can be used as a biomarker, an optical biomarker for Plasmodium parasites in blood. The figure below shows the absorption spectra, optical spectra for hemoglobin in oxygenated form, deoxygenated form, and hemozoin. And the three spectra are distinct. So the disease conventionally is diagnosed by three main methods. That is optical microscopy, which happens to be the gold standard method, lateral flow assay, also known as rapid diagnostic test or RDT, and polymerase chain reaction technique, PCR. Now, these methods have got their limitations, in which include labor intensiveness, low detection sensitivity, and complex instrumentation. Now, recently, literature has reported optical methods aiming to address some of the shortcomings of the conventional methods. And some of the approaches taken include then harmonic generation, laser-light depolarization, magneto-optic technique, and photoacoustic effects. However, these methods also present some challenges, including low detection sensitivity, difficulties in calibrating the systems, and complex instrumentation. So we developed an hypothesis of improving the optical report and optical techniques, and we term our hypothesis as the photothermal-induced optical scattering modulation technique, and our hypothesis entails the following. So we irradiate the sample with an optical beam that we term as the probe beam, which is partly absorbed by the sample, and another portion of it is diffusely reflected through the sample, and eventually it is detected by an optical detector. And when we visualize the detected signal, we expect to get a constant intensity signal if the probe beam, the incident probe beam signal is constant intensity. When we introduce secondary optical beam that we are referring as the pump beam, which is pulsed or intensity modulated in time, and the wavelength of this pump beam is chosen so that it can be highly absorbed by the sample of interest, in this case, imosoin. Then upon detecting the probe beam and ensuring that the pump beam is blocked from reaching the detector, what we find is the probe beam intensity now gets intensity modulated. And the modulation pattern can indicate whether the molecular composition of the sample, and aid in identifying whether the brand sample has got imosoin or not. So onto the problem statement, existing diagnostic methods that are used for routine screening of malaria suffer from low detection sensitivity and specificity. And also the recently reported literature, those methods have not found the clinical translation because of low detection sensitivity and complex instrumentation. So there exists an urgent need for development of a rapid, simple, accurate optical-based malaria diagnostic technique. So the objective of this work was to develop a photothermal-induced optical scattering modulation technique for detection of Plasmodium parasite pigment, that is imosoin, using label-free brand smear samples. The specific objectives were to develop an analytical model for describing the photothermal-induced optical scattering modulation phenomenon, to develop prototypes for detection of photothermal-induced optical scattering modulation signals for malaria-infected blood, to acquire and analyze the photothermal-induced optical scattering signals and identify suitable signal features and techniques for distinguishing imosoin positive and imosoin negative blood samples, and determine the sensitivity and specificity of the technique in malaria diagnosis. So in the methodology, we report the development of analytical model for quantitatively describing the photothermal-induced optical scattering modulation phenomenon, and also the development of the photothermal-induced optical scattering prototypes, and the brand sample preparation procedure, and the signal processing algorithms employed. So in developing the photothermal-induced optical scattering modulation phenomenon, we made use of well-known law called Beer-Lambert's law, which govern optical absorption of light in samples, in absorbing molecules. And Beer-Lambert's law indicates that if you have a beam of light, and let us call it a probe beam, incident on a sample, and a portion of that beam is absorbed by the sample, so that portion of it is transmitted and reaches the detector, then the detected intensity, I3, is given by the product of the incident intensity multiplied by the exponential is to negative power mu sub AX, where mu sub A is optical absorption coefficient of the material, a parameter that tells us how much light has been absorbed by the material, and X is the optical path length of the sample. So if now we introduce a secondary beam of light, we call it excitation beam or pump beam, onto the sample while still maintaining the probe beam. So the pump beam will also be absorbed, and a portion of it will be transmitted from the sample. But then now, if we introduce an optical filter such that the pump beam thus is blocked from reaching the detector, so that only the probe beam gets to the detector, what we get is an intensity that may not necessarily be the same, may equal to the value of the initial detected intensity, and the difference, the reason for the two intensities not being equal could be a change in optical absorption coefficient of the material due to introduction of the pump beam. So in other words, the pump beam alters the optical absorption properties of the sample, so that now the optical absorption coefficient changes, and therefore the detected probe beam intensity is different from the initial intensity. And therefore, if we introduce, or if we change the wavelength of the pump beam, again, we anticipate that the recorded probe beam intensity won't change, but the general form is given by equation 1, 2, and 3. Now, we need to note that optical absorption coefficient is a function of the concentration of the light absorbing molecules and a material property referred to as specific optical molar absorption coefficient. Now, manipulating equation 1, 2, and 3 mathematically leads to equation 4, and the right-hand side of equation 4 is a result we are calling extinction coefficient change ratio, whose value indicates the molecular species present in the sample. So how do we implement this model? So we have developed an algorithm for implementing the model, especially in microscopy imaging. And that is done first by illuminating the sample with a probe beam, right? And acquiring an image, call it I1. Then while the probe beam is on, introducing a pump beam of a given wavelength and acquiring the signal, the pump beam's signal, call it I2, and changing the pump beam wavelength and acquiring another image, call it I3, and now performing these mathematical operations, taking the ratio of I2 to I1 and I3 to I1 and their natural logarithm to end up with the extinction coefficient change ratio image. And this image would then display the different distribution of light absorbing molecules within your sample. So to develop the instrument for acquiring photothermal induced optical scattering modulation signals, we used the shown block diagram where the right source is we used a right emitting diode that emits 3 different distinct wavelengths. The red right, which is in the wavelength of 625 nanometers, green right, which is 540 nanometers, and blue right, which is 460 nanometers. Now, blue light was used as the probe beam, and green and red was used as the pump beam wavelengths. Then this light would pass through the sample, and after passing through the sample, we would filter off the pump beam so that only the probe beam gets into the detector. And then the detector signal is acquired by the computer for further processing. Now, from that general schematic diagram, we developed 3 different types of prototypes. One was the photo-diode type, where the optical detector used was a photon diode, and the right source was right emitting diode emitting 3 distinct wavelengths, red, green, and blue. And so the right source would go through, the right would go through the sample, then we would introduce an optical filter element before the right goes to the detector. Then we would put a transimpedance amplifier to convert the photodiode current to voltage, then take it to a digital oscilloscope for digitization, and then the signal would be acquired by a computer for further processing and visualization. The second prototype was what we are calling photothermal induced optical scattering modulation image sensor type. Here, the right source, again, RGB LED, would illuminate the sample, and then an optical filter element, and then the transmitted beam would go to a webcam, which would capture a video clip of the sample, and the signal would be routed to the computer for processing. Now, the driver circuit would modulate the pump beam signal while keeping the probe beam signal constant intensity. And the final setup we developed was essentially to try to incorporate the photothermal induced optical scattering imaging technique using the conventional optical microscope, and we adopted a microscope, an open source microscope called open fracture microscope. And what we did is we introduced RGB LED so that we could produce the probe and pump beam signals that would illuminate the sample, and then the images would be acquired by a camera that is fitted in the microscope. Then time-lapse images would be acquired and processed for further. So the processing of the microscope images entailed used the following algorithm. So we would acquire the videos and then transform the video frames using discrete Fourier transform. And because the probe beam was modulated in intensity at a frequency of 2 Hertz, while the pump beam was not modulated in intensity, it was constant intensity. And so when we perform the Fourier transform and extract the 2 Hertz signal, we only manage to get the probe beam images and drop the pump beam images. So samples were acquired from human subject upon obtaining ethical clearance, and we visited the health centers and requested the patient confirmed to be malaria positive to donate some peripheral blood, and also we got some blood from human subject confirmed to be malaria negative, and we used the samples to obtain the photothermal induced optical scattering modulation signals, either as home brand or brand smear samples, thick or thin brand smears. So the results, first we tried to get signals that would tell us whether the photothermal induced optical scattering modulation hypothesis is there or not. And the way we did it is we acquire probe beam signals when the pump is not on, and acquire probe beam signal when the pump is illuminating the sample, and we compare those two signals. And what we found is absorption, the sample absorption when we introduce the pump beam significantly reduced, and the red plot shows probe signal when the pump is not on onto the sample, and the black plot shows the absorption of the sample when pump and probe signal are illuminating the sample. The second diagram shows the phase plot of both the pump and pump probe signals, and what we see is the phase spectra is also distinct for the case when the pump and pump probe are illuminating the sample. So that shows that introduction of the pump beam modifies optical absorption of the sample. So the next thing we did was to now take the samples that were malaria infected and non-malaria infected and acquire their photothermal-induced optical scattering signals and compare the magnitude spectra of those two sets of samples. And what we found is when we pump the sample with dendrite, we were able to distinguish all the samples that were malaria infected from all the samples that were non-infected. However, when we were pumping the sample with green light, a few samples, we were not able to find frequency spectrum where all the samples could be isolated that have a given malaria infection status. We went ahead and tried to classify different parasite round levels using the photothermal induced optical scattering modulation for samples with varying immosoin concentration. And what we found is when we take first principal component and second principal component, we could isolate non-immosoin negative samples from immosoin positive samples. And when we take second principal component against that principal component, we could isolate different concentrations of immosoin infected brand. So to automate the process, then we trained two machine learning models, that is the support vector machine and the logistic regression model, and to classify the acquired photothermal-induced optical scattering modulation signals as either infected or non-infected. And what we found was, in general, support vector machine performed better than the logistic regression, and with an accuracy of 70.7% for support vector machine and 64% for logistic regression. And finally, we compared images that were acquired and probe beam right, and the ones that we computed the images after acquiring probe and pump beam using this ratio that we are calling modulation index, which is basically probe pump beam image over probe beam image. And what we found is, whereas the only probe beam or a microscope image captured under white light would not reveal any parasite features, when we do pump probe and get the ratio of those images, we could reveal areas with the parasite regions. Again, so we take those pump, probe, and probe images and perform this mathematical operation, which gives us what we are calling extinction coefficient change ratio. Again, we get surface plots that are showing us regions with the parasites having a very distinct appearance or topography. And this is the line scan image of this surface plot. So in a nutshell, we managed to develop the photothermal induced optical scattering modulation setups and proved the photothermal induced optical scattering hypothesis. And we managed to show that using the technique, we could identify malaria positive samples from malaria negative samples. We found that Fourier transform principal component analysis and support vector machine were techniques that could be used for isolating malaria positive samples from malaria negative samples using the acquired photothermal induced optical scattering modulation signals. The classification accuracy of the technique was found to be 70%, with a sensitivity of 68% and specificity of 72%. And we also managed to develop an analytical model for describing photothermal induced optical scattering phenomenon. Thank you.